Hi everyone, these are anatomy and physiology videos for healthcare students. In this video, we'll walk through epithelial tissues. So epithelial tissues cover and line the body surfaces both inside and out. If an organ system has an external opening, it is lined with epithelial tissue. Also, our ventral body cavity organs, such as the chest cavity where the heart and lungs are housed, the outsides of those organs are also covered with epithelial tissue. So are the digestive organs in the lower abdomen. All of that is covered in line with these epithelial tissues. In this picture, you're looking at epithelial tissue from the yellow line up. We'll talk about what that means in just a moment. So to distinguish an epithelial tissue from any other tissue type, we look for two things. First, that upper free layer or upper free surface is called an apical surface. Apex means the top point. Well, here it means the most upper region of your epithelium, and that is the free surface. Nothing is attached to that free surface. Oftentimes in these tissue pictures, you'll see a white space beyond the apical surface. Now there's a basement membrane here as well. That is the deeper portion of the tissue on which the epithelium will rest, and it serves as a connector between your epithelium and whatever is below it. Oftentimes it is connective tissue, perhaps muscle tissue sitting down below. So just like the basement in a home is the bottommost feature, here the basement membrane is the bottom of that epithelial tissue. Let's talk about how to name epithelial tissues. There's two things we'll look at. One is the number of cell layers. Either you have a single cell layer and it's called simple, or you have multiple cell layers, two or more is the minimum, and that will be called a stratified tissue. The next thing we look at are the cell shapes. We're looking for the majority of the cell shapes to match one pattern, either squamous cells, which contain a flattened nucleus, cuboidal cells, which contain a round nucleus, or columnar cells, which are brick-shaped and contain an elongated nucleus. So we'll look at some different examples as we go through our specific types. This first picture is simple squamous epithelium, and this picture shows lung air sacs. So we're looking for the majority of the cells to fit a pattern, not perfection, because these cells are sliced from a very thin tissue piece using a very thin blade. Then they're fixated on a microscope slide and stained for us to view. So I'm not looking for 100% match of what I consider to be a single layer of cells, but I'm looking for most of the picture to meet that standard. So here in the circle, there are a layer of flattened cells, one single layer, and that is a lung air sac. That kind of is shaped like a grape or perhaps a balloon, and that's where we have diffusion of air gases, oxygen and carbon dioxide across that single layer taking place. So this tissue, because it is only a single layer, is built for diffusion. You're going to find it in places where you need diffusion to occur, such as lung air sacs, across the walls of capillaries, which are only made of simple squamous, serous membranes that cover the outside surface of our organs, such as heart, lungs, digestive organs, and kidney glomeruli are the place where filtration takes place. So we're looking at the need to move items easily across a single cell layer, and we'll do that with simple squamous epithelial tissue. In this picture, we're looking at stratified squamous epithelium, and the arrow is drawn between the apical surface and the basement membrane. I'm going to estimate that between those two regions, I have at least two cell layers. That's why it's a stratified tissue and not a simple tissue. The closer you get to the apical surface, that's where you'll find the most mature, oldest cells. The cells near the basement membrane, that's going to be where the cells undergo mitosis. They have an immature cell shape, and may not give you the true shape of the tissue. So for that reason, always look toward the apical surface to find the truest cell shape. You can tell the cells are more flattened in the apical region. Down toward the basement membrane, they look either cuboidal or even columnar in appearance. So yeah, avoid looking down there where those cells are undergoing mitosis. Some stratified squamous forms the skin of our body, and that is keratinized with the waterproofing protein. Inside our bodies, we can have stratified squamous. However, there it is not keratinized. It is, it is not going to be waterproofed inside the body. We find this tissue in areas where there's abrasion because the multiple layers help protect us from that abrasion. So epidermis of the skin, the outer thinner portion, and inside our bodies, lining entries and exits. So the mouth, the anus, for a female vagina, these areas are going to have a lining of epithelial that is stratified in nature. So here we have that upper apical surface. Notice those flattened cells compared to the deeper cells in the basement membrane region. Yeah, really look toward that apical surface for your truest cell shape. Now we're looking at simple cuboidal epithelial tissue. Here I've outlined a couple of rings of simple cuboidal. And again, we're not looking for perfection, but most of these cells have a round nucleus. I don't really see the cell shape, but if I did, these would be pretty square in appearance. So instead, I'm going to focus on the nucleus shape, which is helping me see a round nucleus. That's going to indicate we have a simple cuboidal tissue. Simple cuboidal is often built for secretion. That's what we do from glands like oil, sweat, mammary glands. We also have this kind of tissue forming our thyroid gland follicles. You'll also find simple cuboidal making tubes in the kidneys. We do call them kidney tubules. That's where we may be reabsorbing or secreting items to either dump those items into urine or reclaim them back into blood. So I wouldn't necessarily expect to have a ring of cells every time you see simple cuboidal, but that is often the case. All right, and take a look at that round-shaped nucleus. All right, so here we're looking at simple columnar, and you'll see up and down hills here of simple columnar epithelial tissue. True to form, this tissue is made of different cell types. That's what a tissue is. In the circle, you're looking at a goblet cell. Goblet cells release mucus. So in the digestive system, mucus helps to trap bacteria. It also helps to lubricate the passage of food. That's one location where you're going to find simple columnar epithelial tissue lining the stomach, small intestine, and large intestine. Look at these elongated nuclei. You may be able to see some of these brick or column-shaped cells, but if you're not able to see them, do look for the elongated nucleus. That is a helpful clue that we have simple columnar epithelium. There's sometimes so much material that it's hard to get this stuff to stick. I try to have a memory word that jogs my memory. So when I'm thinking about simple columnar, I think about digestive system lining. So digestive system lining could include stomach, small intestine, large intestine, even gallbladder ducts. So this is one way to kind of make a snapshot in your brain. You know, you've got digestive system lining, and then you can think later on as you can add to your memory all those specific examples. This one is also found in a female's uterus lining and lining the uterine or fallopian tubes. Now, this tissue is called transitional epithelium, and at first glance, you may look at this and say, you know, hey, that looks kind of like stratified squamous. It kind of does. But upon closer examination, take a look at each individual cell. They are tear-shaped and they point down toward the basement membrane. These cells are built for stretch. So their job is to stretch in the lining of your urinary system to accommodate the passage of urine. So again, I have a memory word, and that is urinary system lining. And of course, ureters, urinary bladder, and urethra all have this as the lining to accommodate for the stretchiness that needs to happen for the passage of urine through. So these are not squamous cells in the apical region, not like what you would see with stratified squamous. As you start looking at more tissues, I would encourage you to lay any similar ones side by side so that you can compare the two. This tissue is pseudostratified ciliated columnar epithelial tissue. That is a mouthful, and for that reason, we often abbreviate it as PSCC. So pseudo means false, stratified means layers. So it is a falsely layered tissue. It does have cilia on the surface. There you can see what looks like a shag rug on that apical surface. And these are columnar shaped cells. Now, every cell touches the basement membrane. In reality, it's a simple tissue. It's only one cell layer thick. But due to the nature of the way the tissue sample is taken and cut, it makes this one look like it's falsely stratified. Some pictures of PSCC, you can actually convince yourself you see distinctive layers, but nope, if you were to follow every single cell back to the basement membrane, everyone touches the basement membrane. So here I've outlined 3 cells back to the basement membrane, and that's how all the tissue is built. So that cilia moves in a whip-like motion to help move along whatever it is, mucus, sperm. It's going to move in a current those items along past this tissue type. So like I mentioned, I use a memory word to help kind of jog my memory. So I remember respiratory and reproductive, 2 R words. That's where you'll find this one. In the respiratory system, trachea and bronchi, those ciliated cells are moving along mucus. In the male reproductive system, moving along sperm. Just a heads up here, as you start learning tissues, I would look for patterns. Also, don't get too tied up with colors because the stains may be changed from tissue to tissue. And if you're learning that they're pink or red, you may get on a quiz or test and it be stained purple or blue. That's enough to blow your mind and completely get you off track. So do not study by colors, rather look at the patterns. And keep in mind on quizzes or exams, those tissues may be oriented in any direction. Don't expect an apical surface to always sit at the top of your picture. It may be at the bottom. It's not meant to confuse you. It just may be the way the tissue was taken, or the way it was sampled, or the way it was placed onto a slide. So be flexible when you're learning tissues to color and the orientation of the picture on the slide. Best of luck when you're learning these tissues. If you have any questions, please leave them in the comments for me. And if you liked this video, please follow my channel. Thanks for watching.